Welcome student to the part 1 of chapter 14 respiration in plants. So let's start. What is respiration? Respiration is in plant is oxidation of food material, that is breaking down of carbon-carbon bonds of complex molecules like glucose within the cell to release energy for ATP synthesis, and this is also known as cellular respiration. The energy which is released in respiration is used for absorption, transportation, movement, reproduction, breathing, etc. The ultimate source of food that is respired is photosynthesis. The compounds that are oxidized during respiration are called respiratory substrates. For example, these are carbohydrates, most common, proteins, fats, and organic acids. The energy which is released is not used directly, but it is used to synthesize ATP. And when energy is needed, ATP is broken down, hence ATP acts as energy currency of the cell. For respiration, plants get oxygen from the surrounding and they give out carbon dioxide. And this exchange of gases takes place through stomata or the lenticels which are present on stems. Stomatas are present on the surface of leaf. Now, why plant doesn't need specialized respiratory organs like the other organisms have? It is because each plant part takes care of its own gaseous exchange, so gas transport is very limited. Gas exchange is very low as compared to that of animals. And the third reason is leaves are adapted for maximum gas exchange during photosynthesis. And during this, oxygen is released within the cells. So this oxygen which is released during the photosynthesis is utilized for respiration. Most living cells have contact with the air, so they are located close to the plant surface. In stems, living cells are organized in thin layers beneath the bark. So they also have the lenticels, so it will help them in exchange of gases. In leaves, stems, and roots, parenchymatous cells are closely packed and they have thin cell wall. They're also interconnected and that help in exchange of gases. A complete combustion of glucose will yield energy, most of which is given out as heat. Here you can see the glucose, which is C6H12O6, will react with oxygen. Oxidation takes place that will lead to release of carbon dioxide, water, and energy. This energy is utilized to synthesize other molecules in the cell. Now, during the glucose catabolism, not all the liberated energy goes out as a heat. Glucose is oxidized in several small steps. It enables some steps to couple released energy to ATP synthesis. Now, during respiration, oxygen is utilized and carbon dioxide, water, and energy is released. Certain organisms are adapted to perform anaerobic respiration because they are adapted to anaerobic conditions. And some are facultative anaerobes, that means they can perform anaerobic respiration as well as aerobic respiration depending on the conditions. If oxygen is provided to them, they'll perform the oxidation, that is aerobic respiration. And if oxygen is not present, then they'll move to the anaerobic respiration. But other organisms are obligate. Obligate, that means those organisms will only perform anaerobic respiration. The first step of respiration is glycolysis, which is also known as EMP pathway, which is common in all the form of organism, irrespective whether it's an aerobic respiration or anaerobic respiration. So let's start with the glycolysis, which is known as EMP pathway. It is partial oxidation of glucose to two molecules of pyruvic acid in absence of oxygen. It occurs in cytoplasm of all living organisms. Its scheme was given by Gustav Amden, Otto Merhoff, and J. Parnas. So, this cycle, this pathway is also known as EMP pathway. In anaerobes, it is the only process in respiration. Now let's start with the glycolysis. In plants, glucose is derived from sucrose, which is the end product of photosynthesis or from storage of carbohydrates. Sucrose is converted into glucose and fructose by an enzyme invertase. These two monosaccharides readily enter into glycolytic pathway. The first step of glycolytic pathway is conversion of glucose into glucose 6-phosphate, which is phosphorylation of glucose. And the enzyme involved in this reaction is hexokinase. It is then isomerized to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are same. Glycolysis, it includes 10 steps under the control of different enzymes. ATP is utilized at 2 steps in the conversion of glucose to glucose 6-phosphate and conversion of fructose 6-phosphate into fructose 1,6-biphosphate. The fructose 1,6-biphosphate is split into dehydroxyacetone phosphate, which is DHAP, and 3-phosphoglyceraldehyde, which is PGAL. PGAL is then oxidized with inorganic phosphate get converted to 1,3-biphosphoglycerate, which is known as BPGA. Now during this, two redox equivalents, that is two hydrogen atoms, are removed from the PGAL and that is transferred to NAD+, which will form NADH plus H+. Now, BPGA becomes 3-phosphoglyceric acid, which is PGA, yielding energy. So, during this reaction, that is conversion of BPGA to PGA, will lead to formation of energy, and this energy is trapped in the form of ATP. The next is ATP is also formed when phosphoenol pyruvate converts to pyruvic acid, that is PEP to pyruvic acid. In glycolysis, 4 ATP molecules are directly synthesized from one molecule of glucose. Now, the pyruvic acid, that is 3-carbon compound pyruvate, is a key product of glycolysis and its metabolic fate depends on the cellular need. Here is entire glycolysis. Let's revise this once again. Glucose to glucose 6-phosphate, and here ATP is utilized. Glucose 6-phosphate will convert into fructose 6-phosphate. Fructose 6-phosphate will further convert into fructose 1,6-biphosphate. Here you can see that 2 molecules of ATPs are utilized in it. This fructose 1,6-biphosphate will split into 3 carbon compounds. One is PGAL, another one is DHAP. This PGAL and DHAP are reversible and that converts into PGAL. This PGAL will undergo to release hydrogen ion to give BPGA. Here NADH is synthesized. BPGA will release phosphate and the energy is trapped in ATP that will lead to the formation of PGA, that is phosphoglyceric acid. This phosphoglyceric acid will convert into 2-phosphoglycerate and finally it's going to convert into PEP. And then the last step of glycolysis is pyruvic acid that leads to the formation of ATP. Further, in different cell, pyruvic acid is handled in 3 ways, either in the formation of lactic acid fermentation, alcoholic fermentation, or aerobic respiration, that is Krebs cycle. Thank you for your attention.